a big family called, can anyone read this word? It's almost there. So it's Enterobacteriaceae. Enterobacteriaceae. So its full name means that first entero is enteric, and bacteria it's actually bacillus. So gram-negative, and they are bacillus rods. So this is a big family of enteric gram-negative bacilli. And okay, so that's okay. They share a lot of common properties. We introduced the common properties of them. All of them are gram-negative bacilli. They are rods. And their natural habitat is the human intestinal tract in our body, human intestinal tract. And also, in this family, it's a big family, real big family. It includes many genera, such as the familiar ones where we know that the genus, can anyone read this? We say E. coli a lot. This stands for the capital E. It's Escherichia, right? Escherichia. And also Shigella, Salmonella, Enterobacter, Klebsiella, and a lot of genera. So it's a big family. And some enteric organisms are part of the normal flora, normal microbiota. They don't cause disease in our digestive tract. But we mentioned that before, such as E. coli, once they move out of the digestive tract, they may cause trouble. But some of those members are regularly pathogenic for humans, such as Salmonella and Shigella. We'll introduce in detail today. Morphology and identification. This big family, they look the same. So all of them are short, gram-negative bacilli, rods. They don't form endospores. So all of them, gram-negative, they don't form endospores. And most of them are motile, okay? Most of them are motile. They can produce flagella. But the exception here is Shigella and Klebsiella. Shigella and Klebsiella, they don't have flagella. So based on this information, we know that we cannot, we cannot distinguish them solely based on their morphology. So after gram staining, you check with a microscope, you actually cannot tell what they are actually, because they are so similar. And if you culture the bacteria, they form circular, convex, smooth colonies with distinct edges. And they are aerobic, or some of them are facultative anaerobic. So they prefer the presence of oxygen. When there is oxygen, they grow better. And all of them don't produce oxidase. So oxidase negative. And within this big family, if you compare their capability to hydrolyze sugar molecules, the carbohydrate molecules, they are different. So based on this information, we can tell some difference. This is the carbohydrate fermentation experiment. So how this experiment carried out? They used a very simple setup in these tubes, okay? The glass tube, okay? If you look carefully, there are actually two tubes. The big one, and inside the tube there is a small one, okay, the small one. But the small tube is upside down in the big tube. And in this tube, you put in different type of sugar solution, carbohydrate solution. You also incorporate the pH indicator. Before the reaction, before you culture, the pH indicator would show the red color, Okay, this is the red color. Then you inoculate the bacteria to let the bacteria grow inside the tube. So the bacteria would ferment the carbohydrate, break down the carbohydrate. And this tube can tell you the information, the products of the fermentation. For example, when E. coli utilize glucose and lactose, it's able to produce acid and gas. When there is acid production, the pH will get lower, so that the pH indicator would turn color from red to yellow. So that's how the pH indicator works. And if there is gas production, no matter what gas it is, if there is gas production, the small tube would collect the gas. So you would find a gas bubble in it. So this bubble means there is gas production. While for Shigella and Salmonella, when they ferment glucose, they can only produce acid. And both of them cannot ferment lactose. So this is a carbohydrate fermentation experiment can give us useful information to help us determine what they actually are. Okay, so that's the growth characteristics. For the antigens, this big family has some common antigens. First of all, this big family are all gram-negative bacteria. Gram-negative means that they should have the outer membrane, so they have the lipopolysaccharide, LPS. So which means that all of them should have the O antigen, the outer part of LPS, that's the O antigen, the polysaccharide antigen. And LPS is resistant to heat and alcohol. You cannot denature it. And LPS molecules can stimulate the immune system to produce IgM antibody. It's only IgM. It does not switch to IgG. And as I mentioned, most of them are motile. They have the flagella structure, so that most of them would have the H antigen, which is located on flagella. And this is due to the protein antigen of flagellin protein. So the H antigen can be denatured by heat or alcohol. And the antibody stimulated by H antigen is IgG, okay? The protein molecule usually stimulates to produce IgG. Another antigen of this big family is the K antigen. K here stands for capsule. It's something covering the bacterial cell. We call it capsule. Sometimes you can actually see the presence of capsule structure if the capsule is thick enough. But sometimes the capsule is just a thin layer. You cannot see the structure under microscope, but it can cover the surface. So it still have the K antigen. Exist on the cell surface, and some of them are just protein molecules, but some of them are polysaccharides. It's not LPS, but only polysaccharides. And the K antigen is also associated with its virulence. Okay, so that's 3 major type of antigens they have. Okay, today we are going to introduce the most important pathogenic bacteria in this big family, including E. coli, Shigella, and Salmonella. Okay, this is E. coli. This is a gram stain of E. coli under light microscope. They look like this. Have you done the second lab gram stain? Not yet? You done? And yeah, in your slide, you probably will see a structure like this. Because when they prepare the bacteria, they have 2 types of bacteria. One is E. coli, and the other one is Staphylococcus, the gram-positive one. And this is E. coli under electron microscope. The color is not real color, it's just pseudo color. It shows the flagella structure, okay, and also the cell. Okay, E. coli, they are motile bacteria because they produce the paratracheous flagella, and they are able to ferment lactose. They have all these three major antigens, O, H, and K. Most of the E. coli are member of the normal flora, so they a big part of the normal flora, intestinal flora. But some E. coli strains can become pathogenic if they gain ability to produce some virulence factor, such as some exotoxins. Okay, here the different type of E. coli. For the normal flora, okay, they don't cause disease once they are in the intestinal tract. But once they travel to other parts of our body, such as they come to the urinary tract, they cause infection. So that's due to the normal flora infection. And they can also cause sepsis and meningitis. So that's due to the normal flora common E. coli infection. Some E. coli can gain the ability to become pathogenic if they can produce some virulence factor. And this is a big, several types we are going to introduce in detail. Okay, the first type of pathogenic E. coli is called enterotoxigenic E. coli. Probably you think it's hard to remember the name. Okay, it's short for ETEC. The last two character, or the two letter, EC means E. coli. That's easy, right? The first one means enter, so it's in the digestive tract. And the only important information is on the second letter, T. T stands for toxigenic. So they are producing toxins, okay, the exotoxins. So ETEC is the common cause of travelers' diarrhea, and also diarrhea in infants in developing countries. So it can cause diarrhea. They adhere to the human intestinal epithelial cell. They are not invasive. They just stay in the lumen, adhere to the surface, okay? And they produce two different types of toxins. Exotoxins, one is heat labile exotoxin, short for LT, and the second one is heat stable. So this toxin can survive at high temperature, short for ST. LT and ST, once during infection, these two exotoxins can get into the epithelial cell, okay, upregulate CAMP and CGMP. Here C stands for cyclic AMP, cyclic GMP. And we know that both of them are the secondary signal molecule, which can, okay, the next step is to activate the kinase molecules. And so that this can cause the intense and prolonged hypersecretion of water and chlorides by the epithelial cells. So there will be like secretion of water and chlorides into the lumen. And also, they inhibit the absorption of sodium. So secretion and also inhibit the absorption of sodium, which means that it will cause large amount of water into the lumen, okay? And those water and sodium cannot be absorbed, and that can cause intense diarrhea. So this is the LT and ST toxins problem, causes intense diarrhea. The second type of E. coli is called EPEC. Okay, you just remember the meaning of P here, pathogenic. pathogenic. This EPEC is an important cause of diarrhea in infants, newborn baby, infants. They produce the bundle-forming pili, and the bundle-forming pili can mediate the tight adherence of the E. coli on the epithelial cell surface. This is the epithelial cell. Well, this is a normal epithelial cell in the lumen with macrovilli. Okay, it's a healthy cell with macrovilli. But with the infection of EPEC, this is due to the EPEC adherence. You can see there the macrovilli is disrupted. So, okay, this is the effacement of the macrovilli. Okay, the result of EPEC infection in infants is severe watery diarrhea, vomiting, and also fever. Okay, this is the EPEC. The third one is the enteroinvasive E. coli, short for EIEC. I means invasive. Do you still remember when we talk about bacterial infection, what's the meaning of invasive, invasion? The pathogen can get into the cell or even spread in our tissue. So that's invasive. So this EIEC produces a disease very similar to Shigella infection. The disease occurs most commonly in children. And see what happened for the E. coli. The E. coli can invade into the intestinal mucosal epithelial cell. They get into the cell. So that's the invasive E. coli. Okay, the next one is EHEC, enterohemorrhagic E. coli. Anyone know the meaning of hemorrhagic? What's the meaning of hemorrhagic? Yes, bleeding. So hemo means blood related. Hemorrhagic means there is bleeding. So that is the E. coli infection which can cause bleeding in different organs, such as in the colon, it causes the hemorrhagic colitis, the inflammation in the colon with bleeding. It can also cause the hemolytic uremic syndrome, short for HUS. It produces a Shigella toxin, the Shigella toxin. And this toxin is able to inhibit protein synthesis. So it inhibits the protein synthesis so that the cell would die, and the EHEC can get into the bloodstream, then get into the kidney, and there it causes a hemolytic uremic syndrome. The two different diseases, both associated with bleeding. So that's why we call it the enterohemorrhagic E. coli. Okay, the last one is called for EAEC. So here A is aggregative, so enteroaggregative E. coli. So there must be some like aggregation. So this is what happened. During infection, the E. coli would form a special pattern on the epithelial surface. Okay, they aggregate together, and each cell look like bricks. So they form a special pattern of aggregation adhere to the human cell. It causes acute and chronic diarrhea, and they can also produce the ST-like toxin, enterotoxin, and hemolysin, EAEC. Okay, you just give you some information on this different type of pathogenic E. coli. You want to know the mechanism, how they cause disease, and the target population they cause disease. Most of them are just infants and little children or travelers. Okay, laboratory test. And we know that E. coli infection can be in different parts. So there are a lot of specimens we can consider, like urine, blood, pus, spinal fluids, sputum, and other material. Based on your patient's symptoms, and you can check the smear, staining, and have a quick check. To culture E. coli, we can use different plate, such as blood agar or other differential medium. There is no single specific therapy for E. coli infection, but you can support your patient. You can also try, different type of antibiotics, but you should run the test to determine the antibiotic sensitivity before you utilize any antibiotic. So that's the E. coli. Okay, the second pathogen we want to introduce is the Shigella. Shigella is, see this picture, it's a slender, okay, slender means thin, and it's also gram-negative rods. And they are facultative anaerobes, but they grow better in the presence of oxygen. So it's a facultative anaerobes. And in this picture, you can see that Shigella does not produce flagella, so it's not motile. And Shigella is able to ferment glucose. Okay, it produces acid but not gas. So that's the carbohydrate fermentation results. And most strains of Shigella do not ferment lactose. But there is some exception of a special species, Shigella sanae species. And that's the only species which can ferment lactose. Okay, here in this table it shows us the important pathogenic Shigella species. We don't have to remember this table. It just gives you some information, just in case you need to check this information in future. Okay, we are talking about the Shigella infection. Here are the pathogenesis of Shigella infection. Shigella infection is almost always limited to the gastrointestinal tract. So it does not cause other parts of our body to cause other parts of infection. And Shigella infection is highly communicable. Does anyone know the meaning of communicable? Highly communicable. It means it's very infectious. A very low number of bacteria could infect a person. So here it says that the infective dose is as low as 1,000 bacteria. 1,000 bacteria is very relatively low number. Does anyone still remember the infective dose of Vibro? We mentioned a little bit. The infection dose of Vibro is around 10 to the 9th. 10 to the 9th, so that means 1 billion bacteria, right? So this is much, much lower. But this is not the lowest I've ever encountered. I used to work on Francisella, the virulent Francisella bacteria. The infective dose can be as low as 1 to 10. So 1 to 10 bacteria could kill a person. So it's very infectious and very virulent. So that is why it was developed as a bioweapon during the Cold War. Shigella could invade the mucosal epithelial cells. Let me show you a picture later. It can invade into the epithelial cells. Shigella produces endotoxin, the LPS, and also it produces the exotoxin, Shiga toxin. Let me show you. So this is the invasion of Shigella. The Shigella from the lumen, first they infect the M cell, okay? Then can be engulfed by macrophages, okay? Then the bacteria could multiply, proliferate in macrophages. After that, large number of bacteria could be released, and then they are able to infect the epithelial cell. Once they get into the epithelial cell, you can see here, each bacteria has a tail, right? Okay, and this tail is the F-actin filament. And similar to the Listeria monocytogenes, Shigella is also able to hijack the F-actin filament so that it forms a filament tail behind the bacteria. And the F-actin filament could push the bacteria to invade the neighboring cell so that the bacteria could invade the neighboring cell and spread. So that causes flagella invasive. The endotoxin, that's the lipopolysaccharide, LPS. It causes inflammation or irritation in the bowel wall, in the intestinal wall. While the exotoxin, the Shiga toxin, can act as both enterotoxin and neurotoxin. As an enterotoxin, it leads to diarrhea similar to that of the EHEC we just mentioned. And it can also inhibit sugar and amino acids absorption. So that's the enterotoxin. As an neurotoxin, it contributes to the extreme severity and fatal nature of Shigella infection and to the central nervous system reactions, such as coma and meningismus. So that's the clinical findings. So that's Shigella toxins. Clinical findings. Shigella infection, the incubation period is relatively short. Do you understand the incubation period? What's the incubation period? Right, that's the time between exposure and disease symptoms. So after infection, till the symptoms comes out. So that's the incubation period. It's only like a couple of days. And symptoms include the abdominal pain. So patient would feel pain in the abdomen, fever, and also watery diarrhea. And after that, or a day or so later, the number of stools increases with less liquid, but often contain mucus and blood. So that causes inflammation and bleeding in the intestine lumen. Bowel movement is accompanied by straining and tenesmus. So that's symptoms of Shigella infection. Shigella infection was a problem for a long time in China, especially decades ago, and because of the poor hygiene condition at that time. And in our department, we have a teacher, another teacher. She worked on Shigella when she was in graduate school. But in recent years, we haven't seen Shigella infection for a long time. There might be some isolated cases, but not popular anymore. Is it still a problem in your country, Shigella infection? It's not common, right? Yeah, so with the improvement of sanitation and people are having a better condition of living, and they have less chance to have Shigella infection. Okay, Shigella diagnosis specimens, we use the fresh stool or in the stool you can find a mucus flax, or you can collect the rectal swab and as a specimen. And we can culture the bacteria with differential medium, such as McConkey or EMB agar, select medium. That's the SS agar, Salmonella Shigella agar, or the triple sugar iron agar. It's also a special design agar for Shigella. This medium, we don't have it in our lab, but in the hospital, they might have it. For many cases of Shigella infection, it's self-limited. So the patient would recover by themselves. Even without any treatment, they can recover by themselves. But if you want help your patient, you can try to find some proper antibiotic to help your patient recover. Okay, the last one is Salmonella. Salmonella is, we heard of it before, right? Salmonella infection is actually not far from our everyday life. Probably you have it in your fridge, Salmonella, because it's a common bacteria. Let me show you later. Okay, Salmonella. Okay, general features of Salmonella. Here you can see the presence of flagella, a lot of flagella. So it's very motile, and it has peritrichous flagella. Peritrichous. Salmonella does not ferment lactose or sucrose. So that's the carbohydrate fermentation results. When it ferments the glucose or mannose, it produces acid and gas. So glucose and mannose, it produces acid and gas. And usually, Salmonella is able to produce hydrogen sulfur. You know this gas? Do you know the smell of this gas? No? I guess you didn't do the experiment in high school with the production of hydrogen sulfur. It's yeah, exactly. If you have the egg stayed at home for a long time, it's rotten, and you can smell that. It's not a pleasant smell. And Salmonella is able to survive in freezing water for a long time. And also, the bacteria, Salmonella, are resistant to some chemicals. And those chemicals can inhibit other bacteria, but cannot inhibit Salmonella. So that, such as the brilliant green, it's a dye, we can incorporate such chemicals in the agar to culture Salmonella. And that can inhibit other bacteria, but allow Salmonella to grow, okay? Okay, here it's a model structure of Salmonella that shows us the important antigens on Salmonella. First, it's a gram-negative bacteria, so On the surface, on the outer membrane, this arrow is not in the right location. It's on the outer membrane. You would find the lipopolysaccharide. So that's the O antigen of Salmonella. And Salmonella have the peritrichous flagella, so you have a lot of flagellum protein, so that's the H antigen. And Salmonella can produce the capsule, and here we don't call it K antigen, but we call the VI antigen. VI here stands for virulence. So this capsule antigen is related to its virulence. We call it VI antigen. It's similar to the K antigen for other bacteria, okay? So that's the major Salmonella antigens. Pathogenesis of Salmonella. Actually, the majority of Salmonella can cause disease in a lot of animals. So they are pathogenic in animals, such as in poultry, pigs, rodents, cattle. So what's the meaning of poultry? Chicken? Yeah, exactly. So chicken, Salmonella can infect the chicken. So in the chicken factory or chicken farm, usually they need to give the chicken vaccine to prevent Salmonella infection. But sometimes, if they don't have enough money to vaccinate the chickens, the chicken can get infected. And not only the chicken, Salmonella can contaminate eggs. Do you like eggs at home? You cook eggs for yourself? Yeah, eggs is a popular food. You can cook it in a lot of ways. You can boil it, you can steam it, or you can fry it. And in different countries, different culture, people cook the egg in different way. And some people, they like thoroughly cooked eggs, the hardened egg, while some people like the half-hardened egg, so it's soft. While some people even, they prefer the raw egg. It's just a liquid raw egg. So that's a problem, because when Salmonella contaminate the eggs, they not only contaminate the surface of the shell, okay? They can even get into the egg. They survive in the egg. So that if your egg is not thoroughly cooked, it's possible that Salmonella can cause trouble for your health. And Salmonella can get into our body usually or always through the oral route, usually with the contaminated food or water. So if you want to avoid any Salmonella infection, you want to drink the clean water, and also you cook your food thoroughly to make sure that Salmonella in it is completely killed. Once a human is infected, Salmonella can cause 3 major types of disease, like the enteric fever, bacteremia with focal lesions, and also enterocolitis. Okay, for the Chinese student, we would introduce all these points in detail, but here we will just introduce the enterocolitis. So that's a colon inflammation. So that's the most common disease of Salmonella infection. Symptoms of enterocolitis include the abdominal pain, diarrhea, nausea and vomiting, low fever, or a swollen and distended abdomen. So different type of symptoms. And okay, for diagnosis, specimen, okay, that's easy to understand. We can collect the stool, the urine. You can also collect the blood because it causes the bacteremia that the salmonella can get into our bloodstream. But when we collect the specimen, we should collect different specimen at different time points, because at different time point of infection, the bacteria can be found in the special specimen. For example, the first week, you want to check the blood, while for the second week of infection, you want to check the urine, while the second and third week, you can check the stool, whether you can find the live bacteria. To culture the bacteria, you can use the differential media like EMB, McConkey, and the selected medium for this is the SS agar, Salmonella shigella agar. And when Salmonella grow on the SS agar, it shows the black colony, while Shigella, okay, here, the colony is quite different. It's not black color. So this is the diagnosis. There is a specific test called widow test. So that's a patient suffering from enteric fever would have some antibody in their serum. So you collect the patient's serum, the patient's blood, and check whether there is antibody, specific antibody in the serum. So you can run the widow test on the rapid slide test. So that's a quick agglutination. You can also run the agglutination in tubes. So that's the quantitative tube agglutination test. Let me show you how it works. So you get the patient's serum, then you make a serodilution of your patient's serum. Then in each tube, you add in the same amount of either O antigen or H antigen into each tube. Mix it well, then incubate it at 37 degrees overnight. And you check the results the next day of agglutination. Okay? And here is how to interpret the results. If the tube that showed agglutination has a titer of more than 100 for O antigen and more than 200 for H antigen, we say that this is widow positive, widow test positive. For example, here, if the agglutination happens in this tube or this tube, for O antigen, we say it's positive. For H antigen, it's higher than 200, we say that it's positive. But if it's positive in this tube, okay, that's okay. That can happen in the negative serum. So this is the widow test. To treat salmonella infection, we can use this different type of antibiotics. You don't have to remember these names, okay? We won't have questions on this. Okay, so that's salmonella infection.